Okay, so when we left off, just a reminder, we were talking about absorption of various types of nutrients and eventually about water, vitamins, minerals, and so on. Okay. So brief recap, we went through carbohydrate digestion. So we know that carbohydrate digestion starts with saliva. So we have salivary amylase. We travel through the stomach into the duodenum, then we run into pancreatic amylase, which continues chemically breaking down our carbohydrates. When we left off, we were talking about digestion proteins. We looked at the fact that we have a number of different types of proteases. Endopeptidases that break up internal bonds and exopeptidases that take off amino acids from the ends of our polypeptides. We know that before we end up with an active endopeptidase or active exopeptidase, it's going to start out as an inactive zymogen. And we looked at the zymogen pepsinogen, which is coming from our chief cells in the gastric pits and then getting activated by the acidity of the stomach. And I think we are on this slide when we left off. Okay. So we have our protein digestion starting in the stomach, but we're not nearly done. So our protein that we've partially digested is mixed into the chyme coming out of the stomach. Then that chyme is getting squeezed from the pylorus into the duodenum. And we're going to go past the part of the small intestine where we have our pancreatic duct and our bile duct coming together and putting our pancreatic juices and our bile into the small intestine. So in that pancreatic juice, we have our pancreatic proteases. So we have trypsinogen, chymotrypsinogen, and procarboxypeptidase coming into the small intestine here. So you'll notice in our image, in our diagram, we have trypsinogen and chymotrypsinogen. That inogen part tells us they're zymogens, tells us that they're not active yet. So what we can see happening in order to activate these, just like we had to activate the pepsinogen into pepsin in the stomach by coming into contact with acid, we have to slightly break up these zymogens to activate them. Okay. So this is where this brush border protease, enterokinase, comes in. So this enterokinase, its job is actually, it is a protease, so it does break down proteins, but the protein that we're thinking of when we're thinking about enterokinase are not the proteins we're eating, but the proteases themselves. So enterokinase is really helping us to activate the other enzymes we need to break down the proteins in the food we eat. So enterokinase is a brush border protease, meaning it's stuck in the wall, stuck in this brush border. So what happens with the enterokinase, that we have our zymogen, trypsinogen coming out in our pancreatic juice. It bumps into the wall, bumps into the brush border where we have the enterokinase. That enterokinase is an enzyme, slightly breaks down the trypsinogen to activate it into trypsin. Then trypsin goes through and activates our other proteases that we need to break down food. So you can see it's here activating chymotrypsinogen into chymotrypsin. And it's also activating procarboxypeptidase. That doesn't end in an inogen, but it's also an inactive form. So it's activating that into carboxypeptidase. So that's kind of how a lot of our brush border enzymes work. We need something to bump into them, then activate something, or in some cases helps digest. Okay. So these are the basic groups of proteases we need in order to digest a protein. So we need the pepsin from the stomach from the previous slide. Just making the whole list, right? Stomach. Then we have our pancreatic proteases that are coming in our pancreatic juice down the pancreatic duct. So trypsin and chymotrypsin happen to be endopeptidases. Carboxypeptidase is an exopeptidase. So they are all three actually breaking down the protein in our food. And for our brush border protease is enterokinase. Its job is activation. And aminopeptidase is another exopeptidase, so it's not in the image. It would be embedded in the wall the same way, because it's still brush border peptidase protease, but it's also involved in breaking down food. So all of these other ones are involved in breaking down food. It's just the enterokinase that's helping us to activate.
And can you see the trypsin and the chymotrypsin is endo or
Endo, yeah. So the first two are endo, these second two are exo, and the last one is activating the previous ones. Yeah. So we have a long list of molecules involved in protein digestion. That's the basic idea of how it works. So now we have our proteins broken up. They're broken up into amino acids. We now have to get them into the cells, right? So what we were looking at in that previous image, we had those proteases in the lumen of the small intestine, right? So they're still in that cavity inside the GI tract, but we want to get them to the bloodstream. Okay. So that process is going to look like basically all the other processes we've seen to pull different molecules into the bloodstream. So if we want to get an amino acid into our body out of the lumen, the main way we're going to do that is through sodium-linked secondary active transport. And that's the important piece from this list, really. So if you're going to prioritize one thing, prioritize the sodium-linked part, which is why we have a picture here of something that looks like a sodium-potassium pump, but this one is actually kind of showing us how we would start with this sodium-potassium pump, and then in the same way we took glucose across, we're going to take amino acids across. So this is just kind of a reminder. Would have been better if we had shown a picture of the actual amino acid one. But it's just the exact same process. We set up a sodium gradient like we set up sodium gradients elsewhere in the body, like we set them up in the kidneys, for example, and then we use that gradient to pull our amino acids through. We're using that gradient, we then end up with a buildup of amino acids inside the cell. So on the other side, we'd have a channel, and we'd be able to use facilitated diffusion down this gradient for the amino acids and move into the bloodstream. We can absorb some dipeptides and tripeptides, so not just single amino acids, but groups of 2 or 3. And this still works by active transport. It's just a slightly different type, and we don't need to look at the details. But what would happen if we did absorb a dipeptide or a tripeptide into these cells lining the lumen is we then break them down into amino acids once they're inside that cell. And so they'd still get into the bloodstream in the form of amino acids. Our takeaways here are that we're trying to get amino acids into the blood. We use sodium gradients usually to fuel that. If we absorb a dipeptide or a tripeptide, we're going to break it down into amino acids before we move it into the blood. Our next group are our lipids. So a typical diet contains maybe about 50 grams per day of lipids, and 90% of that is the form of triglycerides. So our triglycerides are our fats and oils. So we know they have the glycerol backbone, and then they have their 3 fatty acids coming off. So I draw this sort of flag structure with these like waves or lightning bolts coming off. I'm talking about a triglyceride. That's the glycerol backbone here, and those are the fatty acids. So when we think about lipid absorption, really that's what we focus on. We do absorb, we have some phospholipids we might be eating, we have some cholesterol that we're eating, but it's not a significant portion of the lipids that we're taking in. So the problem that we have when we're trying to digest a lipid is that, when you're eating, and you're probably drinking some water or something else with your food, the contents of your GI tract is basically full of water, and lipids are nonpolar, right? So they're nonpolar because they have mainly hydrogen and carbon, right? So they're not going to interact with water, they're going to be hydrophobic. This means they're really difficult to break up because just like we know our phospholipids like to form the cellular membrane, that bipolar membrane, basically because they're trying to escape having those fatty acid tails touch water. The same thing is going to happen with our triglycerides. They're going to like bunch up into fat globules. So they're not going to mix well with the contents of the stomach or the contents of the intestine, and they're going to stay in these big sort of lipid globs. We can't take a whole big fat glob into a cell, they're too big. So these fat droplets pose a problem, and we're going to first need to figure out how to physically break apart a fat droplet in order to even get started breaking down the individual molecules. So that's where the lipases come in, but especially the bile that we saw was coming from the liver and stored in the gallbladder. So the enzyme that actually digests a triglyceride are our lipases, right? So our lipases can come and break off fatty acids basically, right? So they can make like a cut here and a cut here. But that's only helpful if they can actually access the triglyceride in question. Okay, so the lipases are coming from the pancreas, which means that if you're taking account the different enzymes we have from the pancreas, right? We had pancreatic amylase for breaking down carbohydrates. We just saw a lot of pancreatic proteases for breaking down proteins. Now we have pancreatic lipases breaking down lipids. So those different enzymes in the pancreatic juice are really important for pretty much all our digestion. But these lipases are going to have trouble because they have to get to the individual molecules, and we have a fat droplet right now. So we have a big glob of fat. So what we're going to do first is we're going to use the bile salts from the liver to break up our fat droplets into smaller droplets. So we're going to have a big fat droplet, and the bile salts are amphipathic. So remembering that amphipathic means they have polar and non-polar parts. So they're able to interact with both hydrophobic and hydrophilic chemicals. So they're going to be able to basically peel off smaller chunks out of our fat droplet. This means that essentially, right, you look at the yellow outline that I've drawn, right, that was our original surface area that was available for the lipases, right? So the lipases would only be able to attack the triglycerides that are on that yellow line originally. But as I'm drawing these green bubbles, right, there's a lot more green on this picture than there is yellow. So that green now is all the surface area that we have after we've broken up that fat droplet. So now the lipases can access a lot more of the triglycerides. So a lot more of them are exposed. So the bile salts are coming from the liver. The liver makes them out of cholesterol. So They make them out of another lipid, right, which is part of why they are able to be amphipathic. And we've seen this property of cholesterol before. We know cholesterol is partially embedded in our cell membranes, at least in animals, and humans are animals. So this is an important property of cholesterol as well. So bile salts made out of cholesterol in the bile. Bile coming from the liver, maybe it's taking a pit stop stored in the gallbladder, ultimately coming through our common bile duct, joining with the pancreatic duct, and going into our duodenum. So we can emulsify fat using these bile salts. So they're going to be able to break up our fat droplet into smaller pieces, basically. So once our fat droplet is smaller fat globules, we are now going to be able to access more parts of that chunk of lipid that we've eaten. So this is where our pancreatic lipases are going to start to work. So they are able to break up triglycerides into a monoglyceride and fatty acids. So we'll take a look at those on the next image. But basically, they're able to peel off the fatty acids from that glycerol backbone. Now, some of those fatty acids and monoglycerides are going to get absorbed just directly across the membranes of the cells. And they can do that because they're nonpolar, right? So they have that same nonpolar property, that hydrophobic property as a lot of the cell membrane has. So they're able to get across that bipolar cell membrane, move directly in. We don't need any proteins to pull them in. We don't need transporters necessarily. But not all of the fatty acids are going to get absorbed immediately. Some of them are going to form these structures called micelles. So micelle is another shape that lipids can kind of automatically take if you drop them in water. So before, when we talked back one about how lipids can form a bilayer just because of their chemical properties and the way they orient when they touch water, which is a polar molecule. Micelles are another option. So the micelles are basically a bunch of fatty acid tails pointing towards the center in like a little bubble. So almost like a mini cell, the difference here is it's not a bilayer because it's much smaller. So there's nothing in the center. It's just all these tails pointing towards the middle, basically. So some of these fatty acids have been absorbed immediately, and some of them are floating around in this structure called a micelle. And basically, as we absorb fatty acids, the micelles and the fatty acids and monoglycerides that we're absorbing are at equilibrium, which means that like when we absorb a fatty acid, the micelle is going to release another fatty acid, which we can then absorb, and so on. So as we absorb more fatty acids, we release more fatty acids. So here we see a picture of kind of that whole process. Okay. So we're starting out here with, looks like we start with a droplet with some bile salts on it. We see the lipases coming in. We see them latching on here to one of our triglycerides, and it's broken it up into some monoglycerides. And the only difference between the monoglyceride and the fatty acid is that the glycerol backbone has to go somewhere. So a monoglyceride is just that glycerol backbone still stuck to one of the fatty acid chains. Okay. So our monoglycerides and fatty acids are then floating around in the lumen. Some of them pop directly across the membrane into our absorptive cells. Others are forming our micelles. So that's what these are. Right, so you can see that we have the tails pointing inward. You can see that some of these are made up of some phospholipids. So we have some polar heads pointing out. There we see a glycerol backbone pointing out. Important part is just that the fatty acid tails are inward. And as we absorb some of our lipids, we're going to release more of these from the micelle, and then we'll absorb them in the same kind of manner. So this is just happening by diffusion, meaning that as we have more fatty acids, we're building up a concentration in the lumen. We don't have a concentration of fatty acids in the absorptive cells. We have a lower concentration. So they just go down their concentration grade. It isn't taking any energy. We don't have to use a sodium gradient. It's just happening all on its own. Now, once we're in the cells, we are going to do some packaging of these fatty acids and monoglycerides. So they are going to enter the smooth endoplasmic reticulum, which is the endoplasmic reticulum we don't really talk about much, but they're going to basically pull in those fatty acids and monoglycerides. They're going to rebuild triglycerides. So they've had to come through the membrane in these smaller forms, but then we build them back up into their original structures and maybe into some other lipids in the ER. They're then going to get moved into the Golgi apparatus, which is going to repackage them again into these structures called chylomicrons. And chylomicrons are basically a type of lipoprotein. So they're this lipid that we've made in the ER plus some protein parts. And that's important because that means that a property of these lipoproteins is going to be that because of the protein part, they are going to be able to interact a little bit differently with water. So as we start to move them into the body, we're going to see that we actually move them into the lymphatic system. They can interact a little better with the fluid in the lymphatic system. So the chylomicrons are made in the Golgi apparatus, and they're going to get secreted into the interstitial fluid. So this is fluid on the other side of the cell, and then they're going into the lymphatic system. I have a picture of this one. So here we see at the top our fatty acids and monoglycerides going directly across the cell membrane. We went through the smooth ER, turned them back into a triglyceride. And then they move into the Golgi apparatus along with other stuff, other lipids, other proteins, and we turn them into a chylomicron. So this bubble is a chylomicron. So you'll notice it's kind of big. So that chylomicron, it can't fit through a channel, it can't fit through a carrier, can't fit through a protein. So we have to fold off part of the membrane on this basolateral side. So that's the process of exocytosis, is this folding, right, of the membrane, basically. Right, so we have a vesicle joining into this membrane. It's kind of folding off. This vesicle is bursting, kind of like when we release neurotransmitters. And then we're moving not into the bloodstream, but into the lymphatic system. So that's a special property of the lipids, that they're moving into the lymphatic system instead of directly into the bloodstream. Our carbohydrates and our proteins in the form of amino acids are going into the blood, but our lipids are skipping over the blood and they're going into these small structures called lacteals, which is why we mentioned these lacteals when we were looking at the layers of the GI tract. So they're important because our chylomicrons are going into those lacteals and then into the lymph. Your lymphatic system does eventually link back up with your venous blood going to the heart and get pumped back around. So we do get that fluid back into the body and we do get those lipids back into the body, but they're going through this process first. Just a cool side note from your book. So this is not part of our contents of our slides, but if you ever get your cholesterol measured, that's actually another kind of type of lipoprotein. So that's measuring the contents basically of these same kind of structures of the chylomicrons, basically how heavy they are. So you can have high density ones with a lot of protein, of the lipoprotein, or you can have lower density ones that have a lot of lipid and less protein. And that has implications for buildup, basically on the walls of your blood vessels. Ultimately, we do recycle some of the things that we've been using to digest our fats. So specifically these bile salts, right? So the bile salts have been coming through the common bile duct into the duodenum. They've helped us break up our fat droplets into these smaller little globules. And then they're traveling along, they're absorbing the fat that they've helped to break up. By the time the bile salts get to the ileum, we've done all the absorbing that we're going to do. Remember, absorption of nutrients happens in approximately the first 20% of the small intestine. So the ileum is the end of our small intestine. We don't need these bile salts anymore. So they get reabsorbed into the capillaries, which means they go into our venous blood. And at venous blood, because we're in the GI tract, has to go through the portal system before it gets back to the heart. So it has to go back to the liver first. And as that blood goes back, the liver is just going to grab some of those bile salts and reuse them. So it is making bile salts, but it's also reusing bile salts. That brings us to the vitamins, and I don't have a table for you of the vitamins and minerals we're going to go through, but I kind of recommend for the next couple slides when you're studying. Seems like something that would be really much easier to kind of process if you just make a table of like what thing we're talking about, what vitamin, what mineral, and then sort of like what region of the GI tract is involved. But we'll run through them in slide format for today. So when we're thinking about vitamins, some are fat soluble, and some are water soluble. So some basically are non-polar hydrophobic. Those would be the fat soluble ones. The water soluble ones are going to have polar properties. And so we're going to handle them differently when we're thinking about absorbing vitamins. So the fat soluble ones are going to join together with the lipids. They're able to dissolve in the lipid droplets and in the micelle, and we're also able to package them into the chylomicrons. So if we have a fat soluble vitamin, we're just going to use basically the same process as the lipid digestion. So the fat soluble vitamins that you would be thinking about are vitamins A, D, E, and K. For water soluble vitamins, so a big example would be like vitamin C. These are going to go through transport proteins. So basically, we're going to have membrane proteins embedded in the walls of your GI tract that are able to pull through those water soluble vitamins. Vitamin B12 requires another molecule that we talked about before in our gastric pit. So B12 has to be bound to intrinsic factor, which is made by the stomach. So moving on to the minerals. So when we say minerals, we were often talking about the ions that we've talked about in the body. So we're interested in sodium, we're a little bit interested in chloride, interested in potassium. Sodium is usually just kind of coming through with water reabsorption in some of the ways we saw it work in the kidneys. Right, so we know that often we're moving water by moving sodium. So this is kind of what's happening in a lot of places with the sodium. We do active reabsorption of sodium in the jejunum and in the ileum, and then also in the colon. Remembering that one of the main jobs of the colon is to concentrate feces to make it more solid. So that kind of goes with this pairing of sodium and water. Chloride, just going along with the sodium, they're attracted to each other, the positive and the negative charges. Like table salt is sodium chloride, so the reason they're bound together in table salt is they're attracted to each other, right? So it's going along with the sodium. The potassium is mainly getting passively absorbed as well, but we're occasionally going to secrete potassium, specifically when luminal concentrations are very low. So like when there's not a lot of potassium in the stuff moving through your GI tract. And the reason that's a note you might care about is that this means that potassium balance is going to get kind of messed up in cases where you have like extreme diarrhea and you're losing things really quickly. Methods with concentrations in your lumen, so that might be a case where you see a lot of secretion of potassium and therefore you're messing, basically messing with potassium. Bicarbonate pulls back kind of to our acid-base balance. So we know that one of the things we think about when we think about bicarbonate is buffering of hydrogen ions and management of pH. We know that a lot of the fluid that we have coming through into the duodenum in the form of chyme from the stomach is highly acidic. So we are putting a lot of things that include bicarbonate into the duodenum to start to sort of de-acidify that content so it doesn't like burn the rest of your intestines. So we're doing that with bicarbonate and things like mucus, but then we want to reuse that bicarbonate. We don't want to lose a lot of bicarbonate from our body because we know that we need enough bicarbonate in the plasma to keep our blood pH the same. So we're going to absorb bicarbonate ions in later parts of our small intestines, in our jejunum. And then also we're going to do some sort of fiddling with our levels of bicarbonate in the ileum and in the colon. So in the ileum and the colon, we may also be secreting some bicarbonate and trading it for chloride ions. Basically, we're just trying to balance kind of the pH of the contents of the GI tract in similar ways that we balanced the concentrations of, that we balance the pH of the blood. Brings us to calcium. So calcium gets actively absorbed in the duodenum and in the jejunum. One of the ways this is happening is it's binding to a brush border protein, so another one embedded in the wall, specifically called calcium binding protein, not very exciting. So it's going to get transported into the epithelial cells on those cells where we have it binding to the brush border protein. And then there's going to be a calcium pump on the basolateral membrane, which is going to move it out of the cells and into your bloodstream. Finally, I think this is finally, we have iron. So we talked about the need for iron when we talked about hemoglobin and how we need to have iron in our heme group in order to bind oxygen. So some of that iron we were reusing from old red blood cells, but we also need iron in our diet in order to take in more iron. And so we see some familiar, maybe, molecules here. So enterocytes, those are just our specialized epithelial cells in the GI tract. Secrete some transferrin. So transferrin, right, is transferring iron. Whenever we see this F-E-R-R in a word, that should clue us in. We might be talking about iron. So I think Latin root for iron. Not really sure whether it's Latin or Greek, but it does mean iron. Okay, so the transferrin is being secreted into the lumen of the small intestine. It binds onto the iron. So before, I think we just saw this as T iron to imply that we have transferrin bound to iron. That gets bound to a receptor. We then pull the whole sort of complex into the cell through endocytosis. We're basically folding in that portion of the cell membrane, breaking it off as a vesicle into the cell. Some of it is going to get stored as ferritin. So this is kind of that form of iron we saw before. And some of that iron is going to go and continue into the bloodstream, again bound to the transferrin, so in that T iron. next we get to the absorption of water. So the absorption of water, we're going to be much less detailed about this than we were about reabsorption in the kidney, but we do want to understand kind of the basic problem that we have here, which is that there's a lot of water in the GI tract for two reasons. One, you're taking in, so this intake, you're drinking water, hopefully like 2 liters a day, give or take. But you're also secreting a lot of water into the GI tract as you're secreting all these enzymes for digestion. Right? Like the pancreatic juice is actually juice, the gastric juice is juice. There's water kind of mixed in order to let us move this stuff along, let it flow. Right? You can experience this saliva, right? Like it's not just, you know, chunks of amylase. It's not just those enzymes. There's also water that allows everything to move smoothly along. So there's about 7 liters of secretion in the GI tract. Almost, yeah, you have 7 liters of water in all these various secretions per day. So we need to reabsorb some of that water that is in the GI tract. Otherwise, we will have diarrhea, right? We will have very liquid feces. This absorption is a passive process, and it's just going along with the solutes. So I know I went through that pretty quickly, which should indicate to you that I think those were more kind of tack-on topics. I'll focus in on, help us focus in on what parts of that were really important when we do our review. I wanted you to have a sort of brief view of those. We are going to start the control, secretion, and motility packet so that we have enough time to do some of our reproductive system next week. I want to get that short shrift because it is really important for people's lives. But we're also going to try to sort of keep this to a kind of higher level. So when we talk about GI control, we're fundamentally kind of going to be talking about 2 things. We're going to be thinking about when are we producing these various enzymes that we need for digestion, and we're also kind of thinking about how are we moving the contents of, like the physical contents of the food we're eating. So this control, we can think of it as in 3 phases. And when we say phases, we're really just talking about like where the signal is coming from for this aspect of control. So cephalic phase, cephalic means your head. This means that these components of control of the gastrointestinal system are coming from something in your head. Like if you're thinking about food and then suddenly your stomach starts rumbling, that's cephalic phase. If you see or smell or taste some food, those are all organs in your head, right? That is where that signal is coming from. So those would all be part of the cephalic phase of gastrointestinal control. So this is going to require input from the central nervous system, right? Your brain, part of your central nervous system, it's in your head. We're going to need to send impulses down our spinal cord, right, to get to the GI tract. So we'll refer to these as long reflexes. They have to go a long way, basically. We are going to have some more local stimuli that control your GI system as well. And those we're going to separate out, whether we're thinking about signals coming from your stomach itself, the organ, or whether we're thinking about signals coming from the small intestine. So those are like the 3 places where we can sense things about food. Either we're thinking about food, we're smelling food, we're tasting food. Your stomach can physically sense things about the food that's in it. And same thing with your small intestine, it can physically sense things about the food that's in it. So some of those reflexes in the gastric phase from the stomach or the intestinal phase from the small intestine can be longer and involve the central nervous system, but some of them are shorter. We can kind of bypass going up to your brain. We're going to start with saliva. I'm going to think about like why. why does your mouth water, right? And what exactly is happening there? Okay. So a lot of saliva secretion coming from these cephalic phase types of control, right? Because saliva in your mouth, in your head, so that's not super surprising. So things like the taste and the texture of food, as you've physically taken a bite of something, as you are chewing it up, they stimulate receptors in the mouth. And those are both mechanoreceptors, so feeling physical touch, as well as chemoreceptors, so sensing something about the chemical makeup of that food. Okay, so that's kind of more of a taste thing. I would pair it kind of with chemoreceptors. Mechanoreceptors is a bit more about texture. So you sense these things about the food that you've taken a bite of. Those signals get relayed to a salivary center of the medulla, so in your brainstem. And this is going to hook up to the autonomic nervous system. So the autonomic nervous system in the medulla is then going to send out signals back to your mouth in order to regulate saliva secretion. Sight and smell, so seeing food, smelling food would also affect the salivary center. It's not on here, but I suspect thinking about food probably also does, based on my own personal lived experience. So when you have this input and you notice that you are in the presence of food, this is going to stimulate the secretion of saliva. So you're going to produce more saliva. And depending on which part of the autonomic nervous system, so we have the two parts, the parasympathetic and the sympathetic, we're going to create different types of saliva. So parasympathetic activation would create a kind of watery saliva. And it's the sympathetic activation that's going to create this protein-rich, more viscous, sorry, like sticky kind of saliva. I think next we have a flow chart. We will have a flow chart eventually. So we'll pair it all together when we get there. Okay. So now we're also going to have to regulate how much acid we're making in the stomach and how much carcinogen we're secreting in the stomach. This is going to basically, we need the acid to activate pepsinogen into pepsin to help us digest proteins. So we're going to modulate these two things relative to, is there food in our stomach? Is there food going to our stomach? So the parasympathetic nervous system is going to be regulating the acid secretion. And two molecules that are particularly involved in this are gastrin, which is a hormone that we saw that was directly related to our stomach itself. And histamine is also a paracrine signal that's going to help us regulate acid secretion. Pepsinogen secretion is going along with the acid secretion. So like when we want more acid, we also want more pepsinogen. So this is also under those same controls, like the parasympathetics, the gastrin, and the histamine. So if we're thinking about cephalic phase regulation of the acid and the pepsinogen, we're going to see sort of similar stimuli as for the saliva in your mouth. So things that would stimulate you to produce more acid and pepsinogen are things that tell you you are eating or about to eat. So seeing food, tasting food, smelling food, or physically chewing and swallowing alerts your stomach that you have food incoming. You're going to need to digest something. So it's going to want to make acid and it's going to want to make pepsinogen so that you have the available enzymes to break that down, basically. So this is all activated by the parasympathetic nervous system, so the rest and digest part of the nervous system. And the parasympathetic nervous system is also stimulating gastrin secretion, which we saw on that previous slide as one of the signals. That also controls acid and pepsinogen. So we'll pick up here on Monday You do have a brief quiz over the weekend on the content we've covered thus far in the GI system, and some couple of basic questions about the acid-base balance before. I mean, quite basic. There might be like one. So don't forget to do that. And we're